Hello, I am Peter Park, and today I would like to explain how DNA replication occurs by using a personal model of mine. This very simple model shows the general structure of DNA, a double-stranded helix with base pairs that are not shown. Like other organic compounds, DNA is made up of monomers. These monomers are called nucleotides. Here is how a nucleotide looks like in my personal model. As you can see, the nucleotide is made up of three major parts: the phosphate group, the deoxyribose or the sugar, pentose sugar, and the nitrogenous base. These three parts, when combined, form a nucleotide, and these nucleotides are basically what make up DNA. There are four different types of nitrogenous bases in a nucleotide: adenine, which is represented as red in my personal model; thymine, which is represented as gray; guanine, which is represented as white; and cytosine, which is represented as purple. There are two base pairs among these four different types of nitrogenous bases. Adenine and thymine are base pairs, meaning they bond together in the structure of the DNA. Guanine and cytosine are also a base pair, and they bond together in the structure of DNA. Accordingly, the ratio of adenine to thymine and guanine to cytosine are each always 1:1, meaning that the numbers of each type of nucleotide in each base pair are the same. Now, I will show how DNA replication works. Here is a model of DNA. As you can see, it is double-stranded, and it is composed of a sugar phosphate backbone and nitrogenous base pairs. So in the first stage of DNA replication, the DNA molecule unzips and the nucleotide base pairs separate. Then each existing strand of the DNA molecule becomes a template for a new strand. How is this possible? Well, an enzyme called DNA polymerase provides a catalyst to the DNA replication, and it is responsible for bonding free-floating nucleotides and pairing them up with the existing nucleotides in the existing strand of DNA. So, as the DNA polymerase moves base pair the corresponding base pair follows and bonds so that a new strand of DNA is eventually formed. As you have just witnessed, because of the actions of the DNA polymerase, two identical double-stranded DNA molecules have been formed from the initial single DNA molecule. It is important to note that DNA replication is semi-conservative, which means that each DNA molecule contains an original strand and one new strand that is the exact opposite of the original strand.